Okay, so let's get started. We finished. Should we wait? Okay. So we finished sort of talking about ploidy a little bit, and we can discuss this more later if you guys are interested. I want to move now to a few examples of very nice work showing where specific sets of mutations have led to altered phenotypes. And through these examples, I hope you'll see some sort of general patterns coming up. Yeah? So this is an example of these mosquitoes in Kenya and Uganda, where the forest mosquito is the ancestral mosquito, which actually doesn't care for humans at all, right? And then it got domesticated and loves humans more than anything else. So these people tried to figure out what was the cause of this switch from being interested in all other animals except humans and then being interested in humans. Why do these mosquitoes bite humans so much? So I thought maybe it has to do with odors and how we smell. So they measured the odor emission profile. What have I done? Have we entered some chaotic regime of what? Okay. They measured odor emissions from humans versus animals. So these are whatever, mice jackets or something they made them wear, and then they tested what odors are being emitted. And they find that humans emit a lot of this molecule called sulcitone, and benzaldehyde is something that other animals are secreting more. Similarly, you see here humans are emitting much more of the sulcitone, benzaldehyde less so, other animals are way down here, and chicken is way off there, right? And these are the animals that these mosquitoes seem to prefer much more before they switch to humans. So there is a reference test where there's a cage with mosquitoes here, which you can't see, and either a poor human sticks their hand in or a poor chicken gets to have their odors. And they find that these domesticated mosquitoes have a greater preference for humans, whereas all the other mosquitoes, as well as those in Uganda which don't like humans, are all more interested in the chicken odors, okay? And finally, they map the reason why these odors are being detected by the mosquito much more. This is all saying, what do we emit, right? What do the animals give out? But now you have to understand, why does the insect get these odors much better? Turns out there's an odorant receptor, which are receptor molecules on, for example, in your nose, you have odorant receptors that are expressed, on your tongue as well, which will bind to odorant molecules and sort of report to the brain, this is what this odor is that I'm detecting, right? They found mutations in one of these odorant receptor molecules. So you see the domesticated allele is very, very different. This is kind of a cladogram showing you relationships between, sequence differences between all the different alleles. And they find that all the other mosquitoes that live in Uganda or live in Kenya but don't care for humans, all have alleles that are very closely related, but are quite differently related to the ones that do like humans. Right? So there have been mutations in this receptor molecule, in the genes responsible for this receptor molecule, which have led to these mosquitoes preferring to bite humans over other animals. And now they've kind of specialized on this. And so this is a nice example. Explain which? This one? So effectively, it's a kind of a tree where you're showing the branch lengths are amino acid substitutions. So effectively, roughly, this is, let's say, 1 or 2 or maybe 3 amino acids that distinguish these 3 different populations of mosquitoes from, from Kenya or from Uganda, which, difference from each other here, right? The connecting lines, yeah. And then these mosquitoes, A, B, and G, are groups of mosquitoes that do like to bite humans, and they're quite diverged from this group, right? This G1 in particular. And this is an outgroup which they don't talk much about, but this is a different species, and that seems to be more closely related to this group, right? Which is interested in biting humans. And you can see that there are about 10 to 15 amino acid substitutions here, plus another 10 or so here. So that's many, many mutations that have occurred in this lot relative to all the others. Does that make sense? And these mutations then allow these mosquitoes to sort of find humans, and they're interested in biting humans for that reason. Very, very interesting. So this is a situation where a small number of mutations have a large impact and behavioral shifts have happened, which allow a complete sort of dietary change in some sense, right, for this organism. So another example is from these beach mice in Nebraska, in the US. So you have these white sandy areas interspersed with normal darker color sand or soil, and they find two mouse phenotypes here. Okay, one that is sort of better matched, light colored, matched to the sandy area, and the other one which is darker one. What they did is they had these clay models which they made mimicking the color, and they put them out and they measure attack rates, okay, from various predators, and they found that the cryptic, when this guy is in the sand, it's cryptic, the proportion of attacks are lower, and this guy in the darker soil or the darker one in the lighter soil, which is more conspicuous, is attacked much more. Okay, so it's suggesting, and there's a lot more work that has gone on the system for years and years before, that there's a selective advantage to this cryptic coloration and camouflage. Right? So now they wanted to understand what is the genetic basis of this. How did we get these very different phenotypes in a population which is right next to each other? Right? They could be exchanging genes, but they're maintained. This polymorphism is maintained. So this is a recent paper where they mapped sort of in detail the phenotypes, and they find sort of many different aspects of the phenotype which are all important for camouflage. So here's the dorsal ventral boundary. Dorsal is the upper side, ventral is the lower side of the animal. Here's the boundary. They say the dorsal and the ventral color you can see are quite different. They measured actual hair, right, colors using a spectrophotometer, etc. And they measure a tail stripe, whether the tail is striped or not, and the dark phenotypes, as you can see, have a much darker dorsal color, and the ventral color is also a little bit darker, right? And now what they did is, they did a large breeding experiment and they caught lots of different animals which had a mix of all of these different phenotypes. They did genome sequencing, and then they basically asked, throughout different markers that are spaced across the genome, where do we find mutations that correlate with different aspects of this phenotype? Yeah. So I'm not going to explain in detail what's going on there. We can discuss to some extent. I think some of this will be clearer after a couple more lectures from Uma. But let's see what we can make of what these data look like. So what you see on the x-axis is the first plot is showing you, let's say, the entire genome in some sense. Okay, so there are sequences that they've combined. On the y-axis is the likelihood ratio, saying what is the likelihood that that particular position, the polymorphism that they observe at that position, so the mutations across different individuals, how likely are they, how strongly are they correlated with the phenotype that they observe. Okay, so you see there's this big peak here, and this dashed line is some significance threshold that they are considering. This peak suggests that this region is very strongly associated with having the lighter phenotype. Okay. Now they zoom into this, and that's what the second plot is showing. Now within this, there are many, many more peaks, right? And so they've highlighted a few. And now they've zoomed in further into each of these peaks of interest. And what's interesting is each of these correlates strongly with different aspects of this camouflage color. Right? So for example, this set of peaks here corresponds with the tail stripe. This one here corresponds with the dorsoventral boundary. This one correlates with the dorsal brightness, and so on and so forth. So what's the upshot here is that there are different traits that are all part of this overall camouflage, right, or crypsis, which is important for this organism, which has a selective advantage. And all of the mutations that are responsible for this camouflage to have evolved are located in a single gene called agouti. It's for the color, which corresponds very, very strongly with the differentiation between these two color morphs. Yeah? So this is now an example where adaptation has led to the fixation of a set of mutations. Each of these are in the same gene, but they're all relevant for coloration and different body parts, and together the whole package has better crypsis, right? So it's a very, very interesting study. And Uma sent out a paper yesterday, I think, to everybody. And that paper is now a recent follow-up that just came out, where what they've done is they've done big field experiments where they made these mouse enclosures or exclosures in the field, in the sandy area and the dark soil area. let a bunch of mice let loose there and measured under normal circumstances in natural conditions how many of them survived, how many of the light ones survived, how many of the dark ones survived, looked at the ones that survived, what allele frequency changes do they find in the population after whatever X generations in the field, and sort of asked, now can we correlate these different mutations to the survival chance of each morph outside. It's very, very nice work, sort of linking all the way from sets of mutations all the way to natural selection in the wild. So yeah, it's a nice paper. You should read the new one that Uma just sent. Okay, any questions about this? Does everybody intuitively understand what is going on here? Yeah.
So basically, what this is just telling us that if there are
Now you're off.
Basically, this is telling us that if there are differences in the color, these are the positions which are most likely to be responsible for that difference.
Yeah.
This is not telling us has that change occurred. You're saying that the paper which came out is talking about whether these position mutations have come about due to...
No, they're saying, so these mutations all exist in the population already. Right? There's this population of mice. Some of them are dark, some of them are light. They're saying that those exist in the population. What they've done in the new paper is they've asked if we know what allele frequencies are of mice that we put into this habitat, make sure, yeah? Okay. So basically what they've done in the experiment is they've stopped gene flow. And that's the critical thing, right? Because in this, they've sampled mice from natural environments. So you don't know what has gone on. Yeah.
How did they narrow down or correlate a particular variant with a particular phenotype like tail stripe or DB bump?
So they measured all the phenotypes for all the mice that they sequenced, and then they had genome sequence data for the mice.
And all of them belong to a single gene?
Yeah.
Isn't that surprising? I don't know.
Maybe it is. I mean, I don't have a solid intuition for hair color in mammals and why and how many genes should control it. So no, the gene is the same. It's lots of positions in the gene, but it's a single gene. Yeah, Nara. Can you take a mic?
Yeah. So if they are all like controlled by single promoter, like they'll all be transcribed together. But like, won't you need like them to be like spatially segregated on the body at least?
Yes,
like
I would imagine that different. So I think these are all different exons of the same gene. They're not all part of the same.
They won't be transcribed together, you say?
They'll be transcribed together, but they won't be translated together necessarily.
Okay.
Or translated as part of the same
cluster. Okay.
So I would imagine that there is tissue-specific expression which is variable.
Yeah, that's what I was asking about. Okay.
But that I don't know that for a fact. That's my guess. What do you mean everything? Yeah. So they collected a whole bunch, and I think they also did breeding in the lab to generate these intermediates. Okay. Much harder, right? Because when you have species that have already separated, there'll be lots of differences that separate those two species, not just in this one color form. So unless you're working with species that are very recently diverged, this would be a very challenging prospect. So the reason this works for them is because this is a single population which does interbreed and which is in the same habitat, which is not sort of differentiated enough yet, if it ever will. Is that, yeah? So in some sense, this is spatial heterogeneity in the habitat, which is maintaining all this heterogeneity in the genome.
Hello. Yeah. In their natural habitat, do both morphs occur in the sympatry or?
So it's sort of sympatry because the sandy and the soil, the dark soil, are interspersed. That's my understanding. I don't know the system.
And their final goal was to check the gene flow in between the two
Not
populations.
really checking for gene flow. I think they've done all of that work before. I am not as familiar with the rest of the work. Yeah. Wait for 2 seconds, it starts. Yeah. Yeah, so his question is in that previous cladogram for the olfactory allele example, it seemed that the cladogram was based on number of substitutions, amino acid substitutions, not the identity, but actually it's both. It's number and identity that is taken into account when you create those trees, right? So effectively, if A and B are next to each other on the tree, it means that they share much of the amino acids, but a few are different. Versus B and C, C and D, if they are paired up together, then they would share most of the amino acids, but some are different. And then the common ancestor of all four would share a smaller number. Does that make sense? So it is taking into account both. But it's not taking into account which functional ones are relevant or not. That we don't know yet. Any other issues? Okay, good. So that's one example. Then I want to move to a third example of mimicry in butterflies. So this is what's called Batesian mimicry, where you have a toxic, these are wing patterns of butterflies. These are the toxic ones, which are models, which the ones on the left here are mimicking, okay, the mimetic female forms. In this butterfly, there are 3 different forms. They're called Polytus, Theseus, and it doesn't matter the names. But the point is that each of these 3 female forms mimics one model each, okay? And these are toxic butterflies, which predators will avoid. On the left are males, which are non-toxic, okay? Non-mimetic, and also non-toxic. Is everybody clear with this, or should I explain better? I don't think I did. Once more. So with Batesian mimicry, what happens, and this is also seen in other butterflies and snakes, and I think some other fish and some other organisms as well. Effectively, let's say you have a toxic animal, which has bright warning coloration to tell all its predators, don't eat me, I'm toxic, stay away, right? And there are other animals that can mimic their color pattern to get the benefit of being avoided by predators, but they don't pay the cost of actually being toxic, right? Because being toxic means that you have to find the toxin or make your own toxin somehow, okay? So this is Batesian mimicry, and in this system, they wanted to understand how does these 3 different female forms, remember they're all the same species, okay, they're just polymorphic, how do they come up, and also there's male and female differences, right? So they wanted to ask, what is the genetic basis of this? From prior work, it's known that it's beneficial to have these mimetic forms, right? So it's a selective advantage. The question is, what are the genes that are involved? And what they found is this gene called double sex. So for a long time, because there are many different color changes which you can see in the pattern, right? So there's a white blotch here, it has to be patterned just so, there are red blotches in some cases, there's a stripe, many different things. And independently, butterfly breeders had shown that many of these elements are all controlled by different sets of genes. So what people had proposed is for something like mimicry, maybe there's what's called a super gene, okay, which is effectively a bunch of different genes that are next to each other and that are clustered and behave or are controlled like a single gene. So to test that, what they did is similar kinds of association, like I showed you in the previous plot, right? So each of these dots are showing you where you find polymorphism at a particular locus across the genome. And you're again asking what is the likelihood of a particular polymorphism region or polymorphic region being associated with these different phenotypes, okay? And they find that this interval is corresponding to, with a very high likelihood, is corresponding to this gene called DSX or double sex, which interestingly is a regulator of transcription, regulating many different genes, okay? And it's found all over the animal kingdom. It's a developmentally important gene. It's important for sex determination in many animals, so decides whether males or females will behave like males or females, or will have features of males and females in many insects. And it also controls other aspects of insect development and behavior. And they find that this controlling gene is effectively the super gene, right? So it's not that there are many genes that are all responsible for these different wing types. There are many genes which are controlled by this one gene, and there are mutations in this one gene which are responsible for the mimicry evolution, okay? And because there are different proteins that are ultimately made from this one gene, depending on which exons are put together and which exons are used in males versus females and in the 3 different female forms, it ends up producing this large diversity, okay? And so this is an example of regulatory changes, right? Where there's, if anybody has seen Lord of the Rings, one ring to control them all. And that's how this system behaves, as if it's a super gene. There isn't a true super gene, okay? All right, so then one last example of mutations that alter phenotype. This is also a very interesting study system, where there are 2 different insect morphs here again. These are stick insects. You've seen stick insects around? Yeah, they look like sticks. And these 2 different forms are also a case of potential sympatric speciation. They live on these bushes, okay? And one form has a stripe down the middle, a white stripe, and the other one does not. And the one which has a white stripe prefers to hang out on bushes where leaves have this white stripe, okay? So it's very well camouflaged there. And the other one hangs out more on bushes which are dark green, no stripey, okay? So these are then interspersed and they form a mosaic, okay, this population. You can see that in this phylogeny here. So this, the squares are the striped version, so you see them there, and they're all nested and they're all paired with a population of the Cyanothus form, which is the triangular one, right? You see that? So effectively, it's not that all of the squares and all of the triangles are separated in the phylogeny, which means in a given spatial location, as small as this room, you can find patches of bush 1 and bush 2, each of which has a corresponding stick insect hanging out and, you know, pretending to be a leaf and therefore avoiding predation. Okay. So now they wanted to ask what is leading to this differentiation between these 2 forms, right? And so again, they did sequencing, and here's an interesting contrast to what you find earlier, where in the previous cases, they found one genomic region or one locus, one gene where something crazy was going on. Here, this is a similar kind of scan across the genome, and each little line you see is one chromosome, or more or less a chromosome, called a linkage map. And so it's a linkage group 1, 2, 3, 4, and so on and so forth. And these are FST values. You've seen FST and you know what FST is from Uma's talk yesterday. So basically, you're looking at differentiation between the 2 forms. Where is that highest? And where in the genome does it correlate with? And you'll see that there are peaks all over, right? So it suggests that whatever is going on that differentiates these 2, there are many genes involved. So this adaptation is potentially polygenic, okay? And when you zoom in to 2 of these areas, you'll again see that there are multiple peaks. And this, as an example, they've taken one of these peaks and zoomed in further, and you can see there's this sort of region of whatever, 70 odd base pairs where you find much more likelihood of differentiation, right? So the take home from this example is that there are many, many genes which are involved in this particular diversification pattern. Any questions? Okay, so I want to now sort of wrap up all of these different examples and present you some sort of synthesis of this. This is from a recent review paper. Yeah. What do you mean where these regions will be? No, it's not, regions are not polygenic. The adaptation is polygenic, and the genes, polygenic just means different genes are involved, right? So if many genes are involved, they could either be in a close location on the same chromosome, or they could be distributed across chromosomes, across genomes. We don't know that, but from the previous scan, you can see that these potential candidates which differentiate most strongly between the two forms are all over the genome. There are many, many locations where these seemingly important genes are spread. Does that answer your question? Not quite sure what you were. Okay. Any other questions? Okay, good. So now what do we get from all of these studies? One after the other, everybody will keep sequencing and finding some kind of gene, some kind of gene. Exciting part comes when you start synthesizing this together much more, right? Beyond sort of your favorite crow or your favorite lizard or snake or whatever. So what do we find here? The patterns of differentiation, you can have a continuum of differentiation, like I haven't shown you this example in aphids. There are bursts of differentiation in lizards. So if anybody has heard of these Cuban islands, yeah, anolis lizards, where they've differentiated into these different forms or morphotypes, where on each island, the original ancestors sort of differentiated into a tree climber, a trunk hugger, a grass whatever, and so on and so forth, right? So these are bursts of differentiation that happen suddenly due to strong selection to avoid competition. You have in crows a similar case of a few genomic peaks, not many, not one, but a few, in these two groups of crows, two species of crows. In fish, there are genome-wide differences in herring. In terms of genetic architecture, you find across speciation events. In some cases, only one or a few loci, which have a large impact on fitness, are involved. For example, in the stickleback fish, which again I haven't talked about, but here, stickleback fish, these tiny spiny fish, not good to eat, colonized lakes or freshwater lakes multiple independent times during the ice ages. You've heard of the ice ages, from the oceans, right? And they've mapped the genomic changes to a few loci which control the body armor and the spininess, okay, which is different in marine versus freshwater habitats. And so there's a few genes involved. Similarly, in other butterflies, not the ones I showed you, but heliconius butterflies, all of these are toxic. These color pattern changes, again, are known to be controlled by just a few genes in a few places. Courtship songs in crickets and flowering time in maize, on the other hand, is known to be affected by multiple loci, each with a small impact on fitness or survival in different environments. And this is more similar to the stick insect that we just talked about at the end, right? So you can have many different sort of organisms and case studies which fall along this continuum. And so in general, one question that people in population genetics have been obsessed with for a long time is how often does speciation or adaptation or differentiation between populations, how often does it occur through many genes, each of small effect, versus one gene of a huge effect, right? And of course, like most answers in biology, it depends, right? It varies. There's examples of all sorts that you can find. But this remains sort of an interesting area of study because it means, again, that we can make different predictions for the fate of such systems over time, across different landscapes, across different environments, and so on. So now that we have enough, dozens of studies to talk about this, we can start finding out what are the general patterns. Can we make some predictions? Tomorrow, if a new bird species is going to differentiate or speciate in between South India versus the Himalayas, right? Western Ghats versus Himalayas. Can we make any predictions at all, given what we know about these birds, given what we know about the environment, and so on? Okay? So we can start addressing these questions. And in general, they're proposing that we are moving towards this speciation systems biology, where there's a population network, there's some migration linking these different populations. When you look at populations and individuals, these are the important parameters: selection coefficients, growth rates, mutation rates, and effective population sizes. Hopefully you've heard about all of these so far, to some extent at least. And ultimately, these are all influenced by the genes which reside within genomes. Yeah? This is, I like this figure, it's kind of all-encompassing. Here's a broad pattern. Yeah? Okay, so now let's move to recombination. That was the end of mutation. Here is what crossover means in general when we talk about recombination as a way to generate variability as well. So you have your two chromatids, right? let's say these are the alleles that are on each arm of each chromatid. The little a's and the big A's are basically two different alleles of the same gene. Yeah. So you have one copy from your, let's say, father, one from your mother, and they're both duplicated, and you have two of each that are joined. And here's a crossover that's possible now, where instead of having each of them retain the copy that you got from each parent, there's now a mix-up, right? Where big A and big B on this particular chromosome is associated with a small c instead of a big C, and vice versa on this. So now during meiosis, when these sister chromatids separate and they form new gametes, you're going to have two gametes which do not match either parental haplotype. Does that make sense? Everybody with me? This is why recombination is interesting, and it generates new variants in a way, because it mixes up the set or the pool of alleles that you're working with in each gamete. Yeah, so that we will go over this here. Any other questions? Somebody else had a question? Okay, so then what is actually happening in this crossover? This is not sort of magically, randomly switching around. Typically what happens is there's one break in the DNA, okay? So the DNA of one particular sister, so this is one sister chromatid. Imagine this whole thing is like this purple, right? So there's a double-stranded DNA, there's a break, and that break, when it's being repaired, some ends are chewed up. This is what enzymes do in the cell. And when they're repairing, they may use the other sister chromatid, the red one, to start repairing with the template, right? And ultimately, what that means is you might end up with sister chromatids where now the blue one originally has an end which is red, and the red one has an end which is blue. Okay? Make sense? Everybody okay? The detail, when this is more complicated as a process, there are multiple enzymes involved. This may not always happen, but this is effectively what allows the exchange to occur. Yeah? Any questions? So what we then end up having is, if there are interactions, either interactions between different genes, or if there are order effects where is C and who is it next to matters, then it means that we're generating new combinations of alleles and new variants, and therefore this is legit as a new variation. Okay, and it's kind of a bit like a mutation. Okay, so now what does recombination do effectively, and how important is recombination for evolutionary change? In general, this question has been addressed most frequently in the context of sexual versus asexual species. Why are there sexual species? One hypothesis has been that sexual species exist despite paying a large cost of sex because they have access to recombination, right? Because recombination is more likely to occur in sexual species, and this allows for different genetic variants to be sort of sampled. Okay? So there's clear costs and benefits of this, of recombination per se. Many people have also called for separating the effects of sex from recombination, right? Because there are other effects of sex which are not dependent only on recombination, but the fields have been sort of mixed up. We'll consider recombination separately on its own. So if you look at the rates of recombination, you measure what is the chance that two genes will get exchanged, right? Their alleles will get flipped. This has been measured in many different ways in a whole bunch of organisms, and you find that the rates vary quite a lot. Okay, so on the y-axis is log recombination rate, which is centimorgans per million base pairs. Centimorgan is 1% chance that two genes will be separated by a crossover. So effectively, the highest known recombination rates are in fungi and this clade called SAR, which is straminopiles and some other two things that I forgot. Basically, these very, very basal lineages in the tree of life, which are close to fungi. Okay. Basically, there's a whole bunch of stuff that we just don't know enough about, and that's what I say. So they seem to recombine quite a lot compared to things like conifers. Everybody know conifers? Pines, firs. They seem to have very low recombination rates, right? And so one major question has been in the field, why do organisms have such different recombination rates? Because they're all sexual species, by and large, right? So what gives, why are there so many different recombination rates? Is recombination rate something that could evolve? Under what selection pressure can it evolve? What is the mechanism through which it can evolve? what are the consequences of recombination rates, etc. Right? And recombination rates vary not only across organisms, but they're known to vary also within a genome. So different parts of your genome has lower or higher effective recombination rates. This is very similar to our measurements of mutation, where you're not quite sure whether the mutation rates you're measuring are reflecting both mutation and selection, so actual recombination or just selection. Sorry, selection and recombination or just recombination. Yeah, everybody with me? Okay, so here is a sort of list from a review. I apologize for all the text, but I couldn't find a nice figure. But this is sort of interesting because what they've shown here is they've collected all these studies saying when recombination is suppressed, okay, either in specific genomic locations or as a whole in some way, what impact does it have on adaptation and so on. So what they find is adaptive and reproductive isolation traits that map to recombination cold spots. Okay, so these are parts of the genome which we know independently do not show high rates of recombination. And interestingly, there are many case studies where genes that are thought to be associated with strong adaptation or with strong differentiation between populations are in these recombination cold spots, don't recombine as much. It kind of makes sense, right? If recombination is going to measure gene order or allele combinations, if there are 3 genes that need to have certain allele combinations for adaptation to occur, right? When adaptation is polygenic, for example. If you have recombination, you're going to mess up that adaptive association between alleles. Does that make sense? So while recombination can generate raw material for selection to act upon, the flip side is recombination also destroys allele combinations which may have evolved and are adaptive. Right? And so these are case studies where it's clear that it's the latter that's going on. Yeah.
Yeah. So I heard, I think in some class, that there are recombination sites where recombination is possible from, and it just won't happen like arbitrarily across the chromosome, right?
I don't know enough about the molecular biology of where and how recombination can.
I was just asking then, like in a like a mutation that removes a recombinant site between 3 genes that are supposed to be inherited together, will be under like some selection or something.
Yeah. So that's the general take home from this, although it's not clear in every case whether the recombination cold spot means that selection has removed any recombination possibilities or whatever, or is it that there could have been other genes which may have led to adaptation, but they did not fix because they recombined too frequently, right? So it's a chicken egg question. Clear.
Sorry, could you repeat the last part?
So one possibility is that the reason we find these genes in recombination cold spots is because effectively this became a cold spot after the fact, right? After the adaptive alleles were fixed, right? And then there was very strong selection to not have recombination. Or the other way around, where there could have been recombination hotspots, where you also had beneficial adaptive genes or alleles, but they couldn't get fixed because recombination kept messing them up. So these are the ones that got fixed. And it's not clear which of them is true. Sorry, yeah.
Hotspots and the cold spots are relative to kind of stable.
Genome-wide average typically, yeah.
Okay.
Or whatever average people, yeah.
Yeah, so I have a basic question, like how do we measure recombination? So what genotype should we look at, which is a base? What is the control?
What's your baseline?
Yeah,
it's similar to how we are measuring mutation rates, but in this case you need to know something about the phenotype and so on, right? So the earlier studies typically focused on having known associations between 2 different phenotypes. So let's say a red-eyed fly always has, black wing, right? And a white-eyed fly always has yellow wing, some such thing. And so when you know that there is recombination, you start seeing the hybrid phenotypes, right? And that tells you recombination has occurred. And that's kind of how people mapped recombination distances, and they also use that fact to map distances between genes on chromosomes before sequencing was possible. So it's very similar to, yeah, and has all the same caveats as mutation rate estimation. Yeah. Can you get a...
Is there a correlation between recombination rate and divergence time?
I don't know. Good point. I have no idea. We can look this up though, easy enough.
Expect a divergence, newer species would have more recombination rate compared to others because there's more variation.
Because?
There's more variation in a relatively new species.
Why is there more variation in new species?
Because all the alleles haven't been fixed, because some of them will be selected for and then they would get fixed.
Depends on how speciation is happening. So I think there's much more nuance to that, but I think it's an interesting question to ask. I can bring up a hypothesis either way, you know, and justify it. So it'd be interesting to ask if somebody actually measured that. I don't.
Is it possible for a recombination co-spot to turn into a hotspot and vice versa?
In theory, yes, I guess, but it depends on why, what is the mechanism driving recombination rate differences across the genome, right? So like I said, I don't know enough of the molecular biology involved, so you have something? Yeah. Yeah, so the question is the location on the chromosome important to decide recombination rates? The problem is there are correlations there, so it's true that near centromeres you won't find as much recombination, but the reason why that happens is not entirely obvious. As far as I know, there's no clear consensus because many different features of genomes correlate with position along the chromosome. So one simple correlate is heterochromatin tends to have so this is the stuff that is, the part of DNA which is bound up tightly and is not often sort of open for transcription, translation, and so on. That tends to have lower recombination rates. But there's also associations between heterochromatin and GC content within genome. So which may also then again, I mean, there's correlations left and right, but it's not clear causally what is going on. As far as I know, it's not clear yet. Other questions? Okay, yeah.
Species have somehow controlled their recombination rates
I don't want to go there.
to adapt themselves to their...
Not clear at all. And also sort of difficult to, I mean, as a species, can we decide something and make sure that, well, actually we can now, but I mean, a worm is, as far as we know, completely incapable of making such decisions and editing its genome and sort of not, you know, deciding not to have recombination and so on.
Time and evolution has done this for them.
Yeah, so the correlations exist, right? The problem is we don't know what is the causal set of events. So one always has to be very careful with correlations. You know, there's this very tantalizing hypotheses we can come up with, very difficult to prove a causal relationship. But most decidedly, I would say you can safely imagine that most species don't have the wherewithal to decide something and do it. That's not how it works. The way it works is you have random mutations, including recombination, and they are either selected or not, or they drift or not. Yeah. Okay. Any other issues? All right. Okay. So what's also interesting is this case of inversions, which I mentioned to you inversions today morning, right? What are inversions? They flip. And inversions are really, really cool as well. And we're sort of knowing much more about inversions because again, they basically mean when you have an inversion, because the repair process, the crossover process I just showed you earlier, depends on homology or shared sequence similarity between the two chromatids. When you invert, you lose that, right? Does that make sense to everybody? So if you had GGA, GG, TT, whatever, and now you flip the whole thing, you start from TT to whatever, GG, right? Which means that you can't recombine anymore. So inverted regions prevent recombination. And that is fantastic because then that allows divergence to occur very rapidly. And so in the DSX case, the butterfly, the mimicry gene case, that's what has happened, is DSX is part of this inversion. And so therefore, it means that that allele doesn't recombine. Okay, so all the mutations in DSX cannot recombine, cannot be destroyed. All of those mutations are just one solid block. So inversions are known to be associated with adaptation or with reproductive isolation, many cases also. Didn't get the inversions. So let's say this is our chromosome to start with, and now there's been an inversion such that, okay, and now let's say this has happened in one individual in the population, which is now effectively a mutant individual. Now there's no possibility of recombining because you can only recombine if you maintain that same order, okay? But with this situation, there's no recombination possible because the sequence of C and B doesn't match. So no recombination effectively means that this allele, if it manages to spread in the population, if it's advantageous, has no competition from or no brakes that recombination would put on its spread. Everybody with me? Does that make sense? If it's recessive, yeah, but it may eventually become homozygous. If it flips back, yeah, sure. Yeah, but in general, reversions are very, very uncommon. Yeah, but it matters what base pairs, no? So as an example, say the B sequence is, okay, and now in the reversed flipped version, you have A, G, C, C, G, G, A, T. Now for recombination, you need these regions to be similar, otherwise recombination won't work. Okay, so because these regions are quite different from each other, you can't have crossover. Yeah.
Saying that it won't recombine now, it will be just for up to few generations, right? Because then this rarer type will become common, and then we will start to see ACB, let's say from father and mother, and then again the recombination will start again between them, right?
Yeah, so within here, so it's possible, for example, that over time there are C dash, B dash alleles that come up, and yes, they can start recombining. Yeah, yeah, yeah. But effectively, the recombination frequency for quite some time is going to be super reduced, basically zero for a while. And then, yeah. Does that answer your question? Are you okay? Not quite sure. Let's talk a bit later and we'll look at the mechanism in more detail.
Does events like this give rise to supergene families?
Exactly what people have suggested earlier, that inversions can sort of lock supergenes in place. And there are some examples of that happening. Yeah? Okay? Yeah.
That much satisfied with the fact that here it's written that it's associated with higher fitness also.
Mm-hmm.
So how, is it a general statement or just specific to some genes?
No, so it's only, these are specific examples that they're citing. There's more to this table which I didn't put in, but basically in humans, there are some inversions that show signatures of being under positive selection, which are associated with higher fitness. They don't lead to necessarily, because it's hard to do that experimentally in humans, but they're associated strongly with higher fitness. Any other questions? Okay, good. So what about, is recombination actually adaptive in some cases? Are there sort of experimentally, can we show it? And so this is a very nice experiment with E. coli, where they wanted to ask what is the relationship between mutation rate and recombination as sources of variation that could drive adaptation. So what Tim Cooper did here is took F plasmids, which are mediating recombination, horizontal gene transfer in bacteria. Okay, everybody clear what's a plasmid? Anybody who's not? You have to speak up, otherwise I'll just continue. Okay, so plasmids are basically separate pieces of DNA that are separate from the main chromosome. Okay, most bacteria have a single chromosome with maybe some extra circles which are plasmids, which are smaller pieces of DNA typically, and which typically don't have too many genes, just a few. Okay, these F plasmids are known to carry a factor that allows exchange of the plasmid between bacteria. Okay, so typically many bacteria will not just randomly exchange DNA, but the F plasmid dramatically increases the rate at which this DNA exchange can occur. And so he basically had F plasmid, which is the Rec plus, which are the black dots, replicates of that, or the white ones which don't have recombination, they don't have the F plasmid. And you have mutation rate, which is either low or high. The low mutation rate is your normal E. coli, and the high mutation rate is when they've knocked out one of the DNA repair machinery. Okay, so it increases the mutation rate. What they want to do is basically ask, on the background of low versus high mutation rate, what is the relative fitness or relative rate of adaptation given you do or do not have recombination? Everybody clear? Yeah, yeah. Wait. Yeah. Okay, so what he finds is at low mutation rate, there is no difference between fitness of the evolved populations, whether they had recombination or not. But at high mutation rate, the high recombination populations are adapting better. Okay, their fitness is higher. And the difference in relative fitness similarly is higher from ancestor in the higher mutation and high recombination. So when you have a higher mutation rate, it seems it pays to have more recombination, perhaps because adaptive alleles which come up in one cell can quickly spread in the population much faster than what the cell itself happily dividing will allow.
Asexuals generally, because they don't have recombination, they typically have higher mutation rates. They can evolve while adapting high mutation rates, like Lenski lines. Whereas sexual ones or recombination ones have a lower mutation rate, but this seems to be opposite.
Yeah, I'm not entirely sure why.
I know, but I'm just saying,
yeah,
conventional wisdom seems to be that because asexuals don't have any other way.
Problem is it's very hard to experimentally tease apart, like I said earlier, many things that happen when you do or do not have sex and recombination. So typically, for example, growth rates are also lower for sexual species, right? So there are many differences associated with sex, not just recombination.
So let's think of Lenski lines, for example, which we know are asexuals, right? So they have mutators, which are like 1000 or whatever. Yeah. But I don't know of any examples really where we have mutators with highly recombining species.
Mutators of highly recombining. I don't know, there have been many experimental studies.
There's lots, quite a few experiments, but mostly E. coli, and they do get, in the lab at least, mutators. So so I'm saying asexuals generally have high mutation rates while adapting.
But not always. Mutators
not always, but I'm just saying this seems to be saying that higher mutation rates also have high recombination and Isn't that the message of this slide?
I mean, it's saying that when you have more high mutation rates,
Yes.
high mutation rate overall is beneficial to some extent, but it doesn't seem to be actually terribly beneficial here at all, right? But I think what it's saying is that the recombination allows the spread of that mutation to be much faster, a beneficial mutation which is sampled.
I don't understand this, but okay, maybe I'll ask you again.
Yeah. They don't have recombination at all, but what Kavita is saying is that there they have higher mutation rates that seem to be beneficial. So, not quite the opposite, is that they're both acting synergistically in some sense. Yeah. Yeah. Yeah, but that, yeah, we can talk. But the thing is, there are very few such studies where they've asked in one system if you increase mutation rate versus recombination rate. And the reason he put both of them together was to counter the genetic load argument that would come with mutation rate being increased. But we can, let's talk about this paper much more. Very interesting. Yeah. Get a mic. Turn it on.
Okay. So I don't know, Kavita, I don't know if are they correlating mutation rate with recombination rate? I think they're correlating the composite of mutation rate and having recombination with relative fitness, right?
Yeah.
So, but
the point is that higher fitness, the highest fitness, is associated with both high mutation rate and high recombination rate.
Yeah.
Whereas typically the argument has been that if you typically have one or the other.
So if you have high mutation rate, you have high fitness, or if you have high recombination, you have high fitness.
Right. That's the general idea with sexual versus asexual species, right?
So in sexual species, having a low mutation rate is more beneficial.
That's the argument. But I don't know if there have been experimental tests. Yeah.
E. coli, there are 1,000 experiments.
Yeah.
That kind.
But they find high mutation rates.
I know.
But not always either, right?
No, it's not always, but there's some conditions and parameters.
Yeah.
Like for example, recombination rate, you said that only certain, so theory predicts, for example, that there are only critical values below which recombination is useful and so on. So there will be some parameter regimes in which these scenarios are possible. But conventional wisdom, meaning from experiments and theory, is positively correlated. So that's why I'm a little worried about this one.
So there's a relatively recent study again with Lenski lines where they modulated the recombination rate as well. I mean, they had lines, wild type, mutation rate was at wild type, and they modulated the recombination rate by making some mutants of REC or something. And they found that even their mutants with higher recombination rates adapted faster to whatever condition they had given. This came out in 2018, I think.
So in general, it makes similar to mutation rates. If you increase variability in some way or the other, you're going to benefit, whether it's sexual or asexual species. So my point was that I don't know if people have tested that enough to really, any other questions? Yeah, between, a copy of a gene on a plasmid versus the chromosomal copy. Yeah? Okay, so actually the next data figure gets at that. So this is a sporty, don't worry about the name. Basically it's a gene, it's an allele which is beneficial, which shows up in the evolved populations. And they look at the frequency of their allele over time, okay? And what they find is the dashed lines are the ones that have low mutation, low recombination. Sorry, not mutation, recombination. And the solid lines are the ones which have high recombination. And what they find is overall whatever testing they've done, typically that the high recombination lines fix that allele faster. Okay. And the low recombination or no recombination lot will take forever to get to fixation. Okay. Make sense? So effectively recombination is just helping the fixation of the advantageous allele to be much faster. Okay, so recombination can be kind of a nice thing. That's all I had to say about recombination. There's a lot bigger literature. You can go look for it if you're interested. Okay, so I want to finish by talking a bit about phenotypic plasticity, as promised. This is just an example to show how complicated plasticity can be for snakes in general. This is, yeah, not particularly this particular snake, it's just pretty. But if you think of something like growth rate, which is an important trait of an organism, body size as well as embryonic development, for snakes in general, they're all affected by a large number of environmental conditions, which include weather conditions, body temperature, behavioral thermoregulation, prey availability, non-genetic effects, foraging success, blah, 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 right? And so effectively, there's a whole array of environmental or other non-genetic features of an organism and where it's living, which can impact all of these traits which are very important for an organism. Right? This is where plasticity comes in. As I defined it the other day, plasticity is the differential response to different environments. Okay? And this is also called a reaction norm. So plasticity can be beneficial in some cases. There's a specific sort of abstract idea of how this might work. So imagine this is mean fitness or frequency of phenotypic values. Okay, so imagine you can think of this as height or whatever continuous phenotype you're interested in. And let's say this phenotype is plastic. So fitness W in the old environment as a function of phenotype is given by this distribution. Okay, and let's say the organism is now moving to a new environment. In that environment, the fitness function is different. Okay? So phenotypes that have higher values, individuals that have higher phenotypic values have a better fitness in this new environment. Individuals with a lower phenotypic value had higher fitness in the old environment. Now consider the situation where this is the old population distribution in the old environment. Okay, this is the sort of hypothetical possible optimum kind of thing. Okay, but this is where our actual population sits. Okay, if I measure in, I don't know, toad populations, what is the limb length or something, this is what it looks like. And we've, by manipulating toad limb length in some way, we've determined that this is the actual function. Everybody with me? Yeah. Okay, so now this is what the population distribution is. This population, if it is now in this new environment, is going to be pretty much extinct, right? Because it has no individuals pretty much that are going to be able to be anywhere close to the optimum fitness in this new environment. Yeah? But if it has a phenotypic plasticity such that it can move to the phenotypic space where it's within this range of optimal phenotypes through plastic responses, it may avoid extinction at least for some time. If this plasticity is not heritable, then this population is doomed anyway, right? But if the ability to have these plastic responses is heritable, which many times plasticity is heritable, then this population may survive in this new environment. Everybody clear about that? So plasticity is ultimately, in some sense, also a heritable trait, right? The ability to show a plastic response is a heritable trait and can evolve. For a long time, people debated whether it can evolve, whether it can have an evolutionary implication or not. It's clear it can. There are some cases at least where it clearly does work that way. Okay? So now, what are the costs and benefits of plasticity? If you think of a simple relationship between phenotype and fitness, you may have an intermediate optimum. Each dot here is a distinct genotype. Okay? So if you follow this, this is a sort of standard case where for a given genotype, it has a particular phenotype and a particular fitness. Right? Now, if we throw in plasticity in this mix, what's going to happen is, let's say, this square is going to expand into a bigger blob, right? Where depending on its plastic response, it may have different phenotypes and different associated fitness values. With me? Okay, so now let's consider this same scenario where this is your average phenotype, Zj star in environment J. Okay. This Zij is the average mean phenotype of the genotype I in environment J. With me? And that associated fitness of that Zij is this, whatever it is. Yeah. But now, this is the expected average phenotype. This difference between expected fitness and actual fitness may represent plasticity. So let's say given my genotype, I may have a plastic response which changes my phenotype to be, my fitness to be either same as the expectation or a little bit different, right? And depending on that, I'm either closer to the optimum or farther away from it, right? So this is where the cost of plasticity may come in. Depending on which direction your plasticity is taking you in, away from the optimum or closer to the optimum, you may be better off or worse off.
I was thinking that one phenotype should have only one fitness level. No, ma'am? How is that?
For a given phenotype?
Yeah.
No, so this is the expected phenotype.
Expected phenotype?
Sorry, expected fitness. Okay. But your actual fitness may vary, let's say, because you're in the wrong environment or some other different interaction with somebody else and so on and so forth.
Then this function is defined for some environment.
Yeah. Am I missing your question?
Can you just explain it?
Yeah.
What are the values?
Yeah, so Zj is the optimum phenotype. Zij is, yeah, in that environment. Zij is the actual mean phenotype of a given individual or given genotype. And its actual fitness is, its measured fitness is this, but given the plasticity in the phenotype, it may, the expectation of the average measured, let's say you take all individuals of this genotype and you measure their phenotype and their fitness, you'll get a range, and based on that range you find that this is the average, that's the expectation, right? So you may be farther from the expectation or you may be closer to the expectation depending on which direction plasticity is taking you in. So, for example, here is the fitness residual from your expected fitness. How far are you in either direction? And this is the degree of plasticity. If you have a greater plasticity, you may have a lower fitness residual, so you're farther away from your optimum. If you have less plasticity, you may be closer to your optimum. Okay, that's the cost part. And so in general, for any trait in environment 1 versus 2, if this is your fixed genotype, okay, so for a given genotype, you have one value of trait in environment 1 and different value of the trait in environment 2. These will be your fitness isoclines, where your fitness is identical. And this is your optimum, let's say, where you want your optimum phenotype to be this value and this value for the two environments, the same trait in two environments. And your actual fitness for Z1 and Z2, which are two different genotypes, can be here. The expected is here. So effectively, plasticity may allow you to be closer to the optimum or farther away from the optimum, depending on which direction your plasticity takes you. Okay? So I want to show you an example now of where reduced plasticity evolved. This is a scenario where they took cichlid fish, which are predators, and guppies, which naturally lived in a predator full environment, right? So they took from natural streams where the guppies had evolved to deal with this predator. And for 3 or 4 generations, they had two replicate populations of guppies, which were in the lab without predator, okay? Or maybe not lab, maybe in natural streams where there were no predators. And then they took their brains and they measured expression level of a whole bunch of genes. The goal here was to ask how much plasticity is there in gene expression and how much does that plasticity in gene expression in the brain, presumably that's involved in evading predators and blah, blah. How much does that change when you allow selection to act on the fact of no predators being there anymore? Yeah. So this is a sort of principal coordinate plot. Each dot is an individual fish. These are the guppies, and so they're measuring gene expression. So imagine there are thousands of genes that are being expressed in the guppy brain. So giant matrix of 1,000 genes versus all your different guppies. You squish your 1,000 gene expression data into 2 dimensions, which is what is shown here. And you see that if we start off with the natural population of fish with predators, this is where the population stands. Here is the evolved predator-free populations. Both of them converge on a very similar gene expression level, okay, which is interesting. And here is the natural predator-free population, which they use for comparison. Okay, so what they're saying is the evolved lines, I'm not very convinced by this, but what they're saying is the evolved lines are going away from the predator used to predator population, more towards the natural predator-free population. That's the part I'm not convinced by in this paper. But anyway, the point is they diverge, and then they measure the plasticity in these responses. Okay? So basically, how do you measure plasticity? Phenotype is expression level here, right, of various genes. How would you measure plasticity? Yeah, so different environments in general, right? Let's say you're interested in measuring the plasticity for plant growth in different temperatures. You put this do some greenhouse experiments, put the plants in different temperatures and measure growth. Yeah, whatever it is that your state is of interest. You measure how much is the variation in that phenotype, given that the temperatures are different. So what they do then is they measure the degree of plasticity, which is on the x-axis here, the ancestral plasticity in gene expression with versus without predators, and the evolved divergence in gene expression, so between the 2 populations and the ancestor. What they find is a negative relationship. So populations genes which show greater plasticity in the ancestor have lower divergence between the ancestor and the evolved. Whereas genes which showed lower plasticity to begin with in the ancestor are showing greater divergence. So what they're suggesting then is that populations which have more so basically you see that there is a high correlation between plasticity, which is non-adaptive, which doesn't contribute to adaptive divergence, and the degree of divergence. I think this is a bit confusing. Okay. Effectively what they're saying is genes that were very plastic did not seem to be involved in altered expression during divergence. Whereas genes that did not show plasticity were the ones that changed across evolutionary time. Yeah? And so what they're saying here is over time there is a reduction in adaptive plasticity or an increase in non-adaptive plasticity, which again is not something I terribly agree with, but these are the best data we have, by the way. So in terms of measuring evolutionary responses and evolution of plasticity, we don't have very good data. Yeah.
The ancestral population is the same as the natural predator?
The natural predator wala population. With predator, yeah. Any questions? Okay, so plasticity is a little bit difficult to wrap your head around. What I'll do is I'll send a couple of papers that you can try and read or references, and that will help clarify some of these questions. But we can revisit. I'll try also find better examples perhaps and send you of, yeah. Yeah. Because that's what their populations were put into, no? So this group has been working with guppies, natural populations of guppies in Trinidad forever. And so they have mapped out these streams and lakes and measured everything from water quality to flow rate to kitna upar canopy cover hai, nahi hai, everything, right? So they presumably, I would imagine, picked 2 streams or whatever that were most closely matched in every other way except predators are there or not there. They've also done manipulations where they remove predators, like they did it, and then let them sit for many generations, many years. So they maybe have used some of those streams as well. I'm not sure. Yeah, that's the beauty of sort of sticking to one system and just working with it for 30 years or something. Get incredible insight from. Okay. All right. So I'll send some more reading material for plasticity and some better examples perhaps of where things are not as confusing. But that's what we have to say about variability. Okay.
Correct or not, but to simplify, can we say that just because in ancestral population the plasticity was high, so now when
for some set of genes,
for some set of genes, when the plasticity was high for gene expression, then when we put those new population in the new environment, then they do not have to change the expression that much. That's why we see divergence in gene expression less.
Yeah, no, this is why I'm not.
So, but
I'm not sure
it's
what's the cause and what's the correlation.
Yes, it's just saying ki because they were more plastic, so they have a wider range of expression level. So now when we put them in the newer environment, they do not have to change that much.
So what they don't have is they don't know what is adaptive or not necessarily. The reason they're saying these are adaptive changes is they find that those are most similar to the natural predator-free population, and that's why they get the idea that this is adaptive, because these changes in these genes mimic what they see in the natural predator-free population. But it's not great evidence. Okay, yeah. And so today we're done after this tea, right? See you tomorrow?